Okay, let's just have a class, and I will just call your name at the end, and then just get to know you a little bit. So we're going to continue to talk about staining technology, but at the beginning we will be review this gram stain real quick. So gram staining, which is to differentiate the bacteria to two major groups, a gram positive and a gram negative. So the major steps, we can do a review. It is crystal violet, followed by using gram iodine as a mordant to let bacteria staining the crystal violet stronger. And then we do a decoloration using 95% alcohol. And I said this step is very important. It has to be stick to 10 seconds. And at this moment, if it's gram-negative bacteria, will be black, which means no color there, ideally. And when we're going to do the counter stain with saffron. So, after we finish the gram stain, the results should be gram-positive bacteria. It is purple, or we say blue color. And the gram-negative bacteria, which is pink color. Bacteria which is a pink, or we say it is red. And we say very straightforward, gram-positive bacteria for purple and blue, it is actually crystal violet color, and gram-negative bacteria stains pink and red, it is actually saffron color. And then we also mentioned the principles behind it. Why gram-positive will stains purple and blue, and the gram-negative is pink and red? There's two theories. The first theory is cell wall theory. And the cell wall theory, which means gram-positive bacteria has very heavy peptidoglycan. So it will be protect stain, not decolorated by 95% alcohol. So it will be showing the crystal violet color. And the second theory, we call it a lipid theory. And the lipid theory is gram-negative bacteria has a higher or greater amount of lipids because it's from the lipopolysaccharide. So the lipids A section, it is about 10 to 15%, versus gram-positive bacteria only about 5%. So the lipids together with the dye will be easily washed off by 95% alcohol. Therefore, it is counter stain showing saffron color. Saffron color. It is pink and red. Okay. This is what we finished. And we also talk about the gram-positive bacterial cell wall structure and the gram-negative bacterial cell wall structure. And we also want to mention several things. Gram-positive and gram-negative bacteria is just one of the results of the gram stain. For the gram staining, we should record three important results, which is A, gram reaction. This is everybody knows, positive or negative. B, we should record, is morphology. So what is morphology? This is showing you at the end of the slides, you could see. It could be coxal, maybe coxal, maybe it is rod shape. And you can see right here, maybe it's a spiral, and etc. But more important, we also need to know the arrangement. And the arrangement should be a single rods. It could be, you see like two of them stacked together, we call it a diprocoxyl. It could be four of them stacked together, we call it a tetrade. And the slide also tells you it could be 8 of them stacked together. Okay? That means it is like a sausage. And also could be a grape shape, a chain shape. If it's a chain shape like streptococcus, and it is also called like a grape shape, which is a staphylococcus. Grape shape. So these are the things that are very important when you're recording the results. Obviously the lab are not going to be due right now, will be due at the end of the lab because the lab room is used by the other classes. Another thing I also want to mention, because gram-positive or gram-negative staining rely on the cell wall theory, so for gram staining, you have to use a very young culture. What means a very young culture? Less than 48 hours at 35 degrees Celsius. If you more than that, the bacteria will start to die, which means the cell wall is going to start to cracking, and the peptidoglycan will not hold on too much. Okay, so that has to using a very young culture. So this is the section what we talk about is a gram stain. Next, we want to introduce you some other staining technology. This is what we mentioned. The next one is acid-fast stain. First of all, I want to mention gram staining. We call gram-positive or gram-negative. Acid-fast stain, we don't say acid-fast positive or acid-fast negative. If it showed up, we will just say the acid-fast stain results. Acid-fast stain is for very special bacteria causing TB, tuberculosis. This is a Mycobacter tuberculosis. I want to introduce a little bit of else. Mycobacter. Originally, this is coming from in the dairy farms or the soil. It is well caused. If it's only Mycobacter, usually it's caused the bone. So will generate, we call it a hunchback. There's a cartoon movie called about the hunchback. Okay, because the bone of the bone, the calcium is cracking down there. So there is a relatively mild species. Mycobacter tuberculosis will cause TB. So there is something I want to talk about TB. Very interesting. Often the time in the hospital, the tuberculosis symptoms will be mixed with other lung disease. So if you have people had a fever, had a cough, about 3 weeks to 4 weeks, you're going to have to see the doctor. And the doctor will do is an X-ray diagnosis. We'll see a cloudy area in the lung. But this is not very good. Whenever you see this, the first reaction, it could be a lung cancer. Certainly you don't want the lung cancer. So what they do, they will do is a testing. Test of TB, which is called BCG testing, which is a skin test. So which means if you already have a TB, you will see that swelling after 48 hours. However, very interesting, if you have a BCG vaccine, which is a vaccine against TB, you also gonna have that swelling. So this move to the next step. At the same time, we're gonna get you sputum sample to do acid fast stain. Now, why we have to do the acid fast stain? This Microbial tuberculosis has a very waxy cell wall. Why they have a very waxy cell wall? Because they have a very special components which called mycolic acids. It is a very long, sometimes we say short to long chain unsaturated fatty acids. So the mycolic acids makes the tuberculosis a very waxy cell wall. Therefore, general, we say basic dye cannot penetrate. Which means if Microbial back to tuberculosis, you go ahead to do a gram stain. It is very hard to do because crystal violet safranin may not be penetrated. So they have to do an acid fast stain. Now what is acid fast stain? The smear preparation is exactly the same. The difference is you need to add a very special dye called carbofusion. Let me make sure the spell is correct. A very special dye called carbofusion. It is right here. Carbofusion, F-U-C-H. A carbofusion. At the same time, you need to put a heating here for 5 minutes. Okay, so the heating 5 minutes together with the carbon fusion, and we also need to do is a counter stain. The counter stain with methylene blue. Okay, that we have to do the counter stain. After doing this, counter stain with methylene blue. And this is, we call it a heat steaming. We don't call it a heat fixing, we call it a heat steaming. This will let the dye go inside, will be showing you. If it is a bacteria showing the acid fast stain, what do you have to do? This guy is presumptive TB. And then we have to isolate tuberculosis from sputum sample. And this needs to send to reference lab. I think in West Virginia, this reference lab could be in Lexington, Kentucky. In the south part, it is in Atlanta. Go to the reference labs, they will do Pfizer isolation and diagnose. The tricky thing is that tuberculosis, Microbial tuberculosis, this bacteria is a doubling time, which means a generation time, which means 1 become 2, 2 become 4, 4 become 8, is very long. It took about 12 to 24 hours. Some species even 40 hours. So you cannot let the patient stay there, just keep coughing, and then they say, go home for 2 days, let me get isolation of the TB that I determined. That's why we have to do acid-fast stain. If you see acid-fast stain positive, then presumptively TB, tuberculosis, you have to do antibiotics immediately. But right now there is another issue is because we're using the antibiotics too much. So they generate a multi-drug antibiotic resistant TB. That's another issue for these type of things. But in the old time, this acid-fast stain is a very important step with other symptoms to presumptively diagnose TB. It saves a lot of time, lots of the energy to diagnose this, and saves the time for the patients. Okay, so that's acid-fast stain. Next to a couple of things, we want to talk about the endospore stain and also touch about the other things. So let's just move this out. We want to talk about endospore stain. So endospore, we're going to using a green color because that's related to another dye. Endospore stain. First of all, we want to mention, not all the bacteria has endospore. There is basically 2 genomes has endospore. It is Bacillus and Clostridium. Bacillus, we usually talk about Bacillus subtilis, cereus. Clostridium is perfringens, tetany, botulinum, difficile. There's different things we'll talk about later. But from the genome standpoint, is Bacillus and the Clostridium has endospore. E. coli does not have endospore. Salmonella does not have endospore. So those we call non-endospore forming bacteria. Bacillus, Clostridium, we call it an endospore forming bacteria. Second, the endospore, it is a protective structure. Usually, it is in a very stress, or we say it is in a harsh environment for bacteria to survive. Let's say you have a acid environment, you have a UV light, you have a little bit higher temperature. It's a protective structure. Now, why we need to do the endospore stain? Because the location of the endospore is genetic dependent. So there's lots of research has been showing if some of the Clostridium, that could be center, the location. And some of the Bacillus, that could be at the end, we call it a terminal, the location. And some of other bacteria, that could be the between center and the terminal, we call it subterminal. And if you know it's going to be one way or the other, either Bacillus or Clostridium, then you do the endospore stain, you know the location of the endospore, you maybe can guess which strains it is. So it's a location is genetic dependence. Now, last thing we also want to mention, why this endospore is a protection structure. The outside, you have a cortex and a coat. Okay, you have a cell wall. This is very similar to the cell wall structure, composed by peptidoglycan. But this inside area is the key. There are 2 major chemicals will help bacteria endospore to survive in a very harsh environment. It is calcium diproconic acids, and this acronym we just say calcium BPA, and has a small acid soluble protein. These two chemicals there, DNA protein, will let the bacteria called protection from the harsh environments. And I also forgot to mention this is a protective structure, so there is no reproductive function, which means the mother cell has an endospore does not mean it will be automatically transferred to the daughter cell unless the daughter cell is also in a very harsh environment. Because it's a protection structure, it's not a reproductive structure. The endospore, if they generate endospore, we call it a sporation. If the sporation, when the condition changes, the endospore could go back to the vegetative cells, we call it a germination. Some of the endospore could be there forever, for the rest of your lifetime. Okay, and regarding the big story behind it, we will talk in the exam too, when we go talk about the disease. Now, how to do the endospore stain? We had a smear preparation. It's the same thing. Air dry, heat fixing, it's the same. When we move on to the next stage, after heat fixing, what we have to do, the smear is there. You need to add a very special dye. It looks like very green, flooding with malachite green. A very green dye. The flooding, which means you basically have to be covered the whole glass slides. Like a flooding happens in your house. The whole area is covered with the dye. And then what we do, very similar to the acid fast stain, we gotta do the heat steaming for 5 minutes. And the malachite green will go inside. Now, of course, we cannot let the slides look like it's all green color. So we do a counter stain using saffron. So what is it going to be looks like? The endospore. The endospore on the glass slides most of the time, you either see a big greenish bubble, like here. Can you see the greenish bubble here on the slides? That's endospore. And the other area, most likely it's red or pink. Or you see a big chunk of the green there. A green dot there. That's endospore. It's very interesting. When you see it, it's fabulous. Now, something we want to say is that gram stain, we just say, the theory is for cell wall. So we need to use a very fresh culture, a young culture, like 24 hours. But endospore, because it is a protection structure in the very harsh stress environments, so we usually use a very old culture. Because we want the endospore has already been formed. Usually more than 72-hour culture is really good. But you know, we did it in the lab a couple of times. If we have a Bacillus subtilis, if we're using Bacillus subtilis, that took maybe only 48 hours to have a good endospore, but Bacillus cereus, serous, they're going to be talking about more than 72 to even 96 hours, couple of days. Even some strains could talk about a week to have the endospore. Okay, so that's endospore stain. Next one, we want to talk about the capsule stain. So the capsule stain. Capsule stain, we want to mention you, is a very special bacteria. It is Streptococcus pneumoniae. And you see outside is a heavy capsule. This is by polysaccharides. This is have an anti-figose structure. So it's SO-ME, which means the pathogen is going to your body. You have those macrophages want to attacking it. It's a protection structure there. So capsule stain, how we do it? There is two methods we can do. One, using negative stain. Negative stain you can use in Indian ink and nigrosin to stain in background. And you will be showing the capsule is well showed up. So you can see a good example is right here. That's a negative stain, the capsule which is showed up. But you also could do is a directive stain. The directive stain you could using copper sulfate to do a directive stain. Directive stain the capsule, but it is a little bit difficult to do. Okay, not using too often, but negative stains we using that lots of the time. Okay, flagella stain. The next one. Flagella stain, we want to mention several things. Flagella coming from protein called flagellum. This is a difference between gram-positive or between bacteria and the eukaryotes. Eukaryotes, it is very interesting, which is logs like 1, 2, 3, 4, 5, 6, 7, 8, 9, like that. If you cut a piece of their flagella. So for eukaryotes, we call it a 9+2 structure. But we don't want to mention here. For bacteria, there are 4 different types of the flagella. Very interesting, because flagella is a structure for real movement, not tumbling. It could be like a tail on the ends of a bacteria. We call it monotrichous. It could be on the ends, but at the 2 sides of the ends, each have one. Amphitrichous. It could be screwed up on one side, but the other side they do not have it. Lithotrichous. It could be covers the whole surface. Petritrichous. Some of the bacteria looks like that. Let's say Proteus vulgaris, a bacteria from the soil. What happened is, on the agar plates, this bacteria will fill a swarm. which means it doesn't even have a colony there because it goes everywhere. So that's a flagella. So flagella, how we do it? We need to stay sick. So how we do? We need 2 different glass slides, like that. And first of all, we need let them rolling very gentle. Rolling like that. And then the top glass slides is very gentle, you repair it. But don't push it too much, very gentle to repair it. To let them be gentle later. And we cannot just use a basic dye to do. We cannot do heat fixing because this is a protein. It will be very easy to hydrolyzed or like melted. You have to add formalin to fix it, to do the fixing. The formalin, you do the fixing, then you use microscope directly to do the observation, you can see it. You can't just use that because it's very, very soft. Okay? So that's something we do. Look at here, the flagella stem. You can see here at the bottom, that's formalin, which is there, and the epoxy. A little bit shiny, they add a fluorescence dye. And this slide, which can show you some of the picture. This is a gram stain, obviously gram positive. You see the purple? Gram negative is pink. Second picture is acid fast stain. You look at these type of color. That's called fusion, and the background is methylene blue. This red one is acid fast stain. So in the exam, I usually ask a question. I said, if there's a patient who is acid fast stain positive, does that mean it's have a TB? The obvious answer should be no, but it's presumptively positive. So you have to use antibiotics. If you see a picture like that from a sputum sample, obviously this guy very possible has TB. This is the endospore stain. Bacillus, look at that. The endospore right there. This is a capsule stain the background. This is flagella stain. This bacterial Proteus vulgaris is the same thing what I just wrote here. The swamp, Proteus vulgaris. It's a bacteria originally from the soil. It's also a very good bacteria for doing the medical lab because most of the biochemistry test, virality test, endospore test, and we had a 3-tube fermentation alcohol test. All kinds of environment, all kinds of biochemistry test, it is positive. So even the motility test, it is positive. So it's a very good tool, very good string for the medical lab because everything showed up positive. It can be do a comparison with the negative results. Okay, so that is very interesting when you see all these type of the stain. And the flagella stain, sometimes you add a formalin to fix this. We also could add tannic acids. If you really want to make it be stronger, we add the tannic acids to make it be more strong. Okay, so that's something we want to talk. And this is, we also show you something about the shape and arrangement. We talk some of here. Look at the picture here, the coccale in the chain. This is Streptococcus agalactiae. We will mention this later on. This is group B Streptococcus. Very important for pregnant women because if they do not do anything to prevent this pathogen, in the later stages of the pregnant, This pathogen could be from the fetus and go to the newborn babies. The newborn baby could have fever, meningitis, and bacteremia, which means the bacteria could in the blood. So Streptococcus agalactiae is very important. Staphylococcus aureus, it's a grape shape, and we will mention that later on. This is a very toxication material actually in the chicken salads, but people doesn't realize it because you do the sneezing like that, and they make the chicken salads. They don't wear the gloves, but they like this, the coughing and sneezing. It's very easy to be transferred into the chicken salads, and you're gonna have a vomiting, diarrhea, real quick, about 3 to 6 hours. The onset time is very short. And although this pathogen we talk about all the time, but CDC does not require you to report it. It's kind of weird to me. Maybe it's too much, but the death rate is not very high. This is the Vibrio chloride. It's a common shape. This is usually in the jellyfish and in the seafood. Campylobacter jejuni, we will mention the chicken section. Campylobacter jejuni is a very strange bacteria. You're using normal bacterial medium, let's say nutrient broth, it's not going to grow. Very weird. They need a very high nutrition to grow. You need to add vitamin A, the blood, and with all kinds of nutrients there. And then even need a very strange environment, which is microallophilic, which means they don't like oxygen. They like carbon dioxide. It's very weird. However, it is a big risk for the poultry meats, but it is difficult to grow in the lab. So it's very ironic. We will mention later on. This one, you see the chain here, Bacillus bacterium. This is usually, we call it a surrogate bacteria for Bacillus subtilis, which means it's not that toxication, and it's showing the same characteristics with this pathogen. So we do this in the lab sometimes to do the observation. This is also going to generate endosperm. Leptospira interrogans. It's a spiral sheet, and we call it a hook shape. And this picture is very obviously taken from dark field microscope. Look at the background is dark, and the specimen which showed up. And this is spiral sheets. Same category with syphilis, Treponema palladium, and Lyme disease, Borrelia burgdorferi. So it's very interesting that one. When you see the whole hook shape like that, Leptospira interrogans, it's a spiral sheets. Okay, so this section we finished. We talk about staining technology. Pay much attention to the gram stain. Absolutely have some question will be for the gram stain. But it's open book, you can look, but you still need to know the principles behind it. Okay, gram stain is a major part of this section. So we're gonna move on to the lecture 4. We want to talk about is the bacterial growth. It's very interesting. We're going to move on to the next section for the bacterial growth. Finish talk about these staining. We want to talk about how the bacteria are going to grow. And we also want to talk about factors to impact a bacterial growth. So these are the things we really want to mention and really want to talk. So we moved here a little bit. At least in this space we need it. We want to talk about that in detail. Bacteria grow. Very important. We call binary diffusion. Binary diffusion. Binary fission. Fission. binary fission. Said very simple. Let's just draw. Easy. This is the mother cell. Become two daughter cells. These two daughter cells go here. Become another two daughter cells. Very interesting. Each like that. So, what you see? This is 1. This is 2. This is 4. This is 8. That's very interesting. We're using 2 to the power of 0 is the mom. 2 to the power of 1 equals 2 is the first generation. 2 to the power of 2 equals 4 is the second generation. 2 to the power of 3 equals 8 is the third generation. So basically what you're going to see, 16, 32, 64, 128, 216 or 215, 512, and to the power of 10, 1024. That's binary fission. And very simple, 1 becomes 2. And ideally, if you look at the slides, the genetic information from the mother cell to the daughter cell should be exactly transmitted. Of course, going through is a central dogma, which is DNA, replication, transcription, translation. In the assumption there is no mutation happens, the daughter cell should be exactly the same as the mother cell. Although it never happens, more or less, they have some of the mutations. Okay? Binary fission, it looks like that. If you explain by the mass, it looks like this. Therefore, it comes out with a very important concept called generation time. We also call it a doubling time. Doubling time, which means how long it needs to take from 1 to 2. That's called generation time. And here the slides you can see, E. coli is very short. Every 18, 20 minutes we'll have 1 generation. So let's say we had an E. coli, just a generic E. coli. We grow 37 degrees Celsius 24 hours. It could from 1 cell to 10 to the 8 to 9th power cells per ml. Because the generation time is very short. But how about the microbacterial tuberculosis? We just mentioned that. The TB strain, 12 to 24 hours is just 1 becomes 2. So how many days do you need it? Sometimes a week you need to do the cultivation. If you have a sputum sample, cultivating to the transport media, go to the reference lab, it took a week to let them grow. Because it's very difficult to detect if you only have 1 cell or 2 cells. At least you need to have like 50, 100 cells and you can do a diagnosis. So here you can see the major difference between microbacteria, tuberculosis, and E. coli. Therefore, it comes out with a very important curve, bacterial growth curve. It is very interesting. Lots of you have another assumption. Okay, let's draw a picture. whenever you draw, you need to label the x-axis and the y-axis. What are they? This is a common mistake lots of you will be make during the exam. Even it's a very easy question you could copy. Lots of people just draw two arrows. It's wrong. What is the unit of the x-axis? Obviously, it's time. That could be second, that could be minute, that could be hours, even days, but at least you need to write a time there. What is the units of the y-axis? It is a number, which means it's the population. So what's that? Cells per ml, usually. Or CFU per ml, colony forming units per ml. Now what's a curve looks like? Lots of people think that goes like this. No, it's not like that. It's not linear. Otherwise, you can imagine I grow a bacteria in a broth for a week. It's going to be more than the human being in the whole world. It's not really like that. What it looks like. The curve is very interesting. It looks like this. And not really. They give you something a little bit very flat at the beginning. Can you see it? This looks like this. So, this is the bacterial growth curve, have 4 stages. First stage, lag phase time. At this stage, bacteria is due to preparation. Their DNA, RNA, their proteins, everything is due to preparation. Especially as they go to the new environments, they need to accustom to the new environments. So, they did not really grow at a certain stage. Once they are accustomed to the environments, they got to grow rapidly. It's very sharp and steep there. This is called log phase, which means they grow dramatically. And you also could see the doubling time, majority is in this log phase.